Good morning everyone. Today we are going to talk about development and histology of parotid salivary glands. So we know there are three major salivary glands consisting of parotid, submandibular and sublingual glands, and there are numerous minor salivary glands distributed throughout the oral cavity within the mucosa and submucosa. And on average about 0.5 liters of saliva is produced each day, but the rate varies throughout the day. That is, at rest 0.3 ml per minute is produced, but this rises to 2 ml per minute with stimulation. So at rest the parotid gland produces 20%, whereas the submandibular gland 65% and the sublingual and the minor glands 15% of saliva. Parotid secretions are almost serous. The submandibular secretions are mixed and the sublingual and minor salivary secretions are predominantly mucinous. So we know saliva is of two types, the serous secretion as well as mucinous secretion. So parotid gland, now we are going to talk about parotid gland, which is mostly serous secretion. So what is saliva? Saliva is essential for mucosal lubrication, speech and swallowing. It also performs an essential buffering role that influences demineralization of teeth as part of the caries process. So when there is a demineralization, it happens in an acidic environment. To buffer that, saliva and its constituents are helping in the caries process. So when there is a marked deficiency in saliva protection, that is xerostomia, rampant caries and destructive periodontal diseases occur. So the protective role of saliva is lost in xerostomia, which leads to the destruction of the teeth as well as the periodontal tissues. And various digestive enzymes like salivary amylase and antimicrobial agents like IgA, immunoglobulin A, lysozyme and lactoferrin are also secreted with the saliva. So these are all the constituents of saliva in general. And when we see the embryology, how does the parotid gland develop? The parotid gland develops as a thickening of the epithelium in the cheek of the oral cavity in the 15 mm CRL embryo, that is crown rump length embryo, on the epithelium of the cheek. This thickening extends backwards towards the ear in a plane superficial to the developing facial nerve. So we know parotid gland is associated with the facial nerve. So it travels back there. And the deep aspect of the developing parotid gland produces a bud-like projection between the branches of the facial nerve in the 3rd month of intrauterine life. And by the 6th month of the intrauterine life, the gland is completely canalized, which means that canals are formed. A bilobe structure, the parotid comes to form a larger 80% superficial lobe and a smaller deep lobe joined by an isthmus between the two major divisions of the facial nerve. So the facial nerve forms a structure in between the superficial lobe and the deep lobe. And the branches of the facial nerve lies between these lobes, invested in the loose connective tissue. So always the parotid gland is associated with the facial nerve and its branches. So in the anatomy, if we see, the gland is situated in the space between the posterior border of the mandibular ramus. We can see the parotid gland and the facial nerve. And the mastoid process of the temporal lobe. The mastoid process and the posterior border of the ramus. The gland is surrounded by a fibrous capsule, which is the part of the superficial mucoacroneurotic system (SMAS), and it is a triangular with apex directed interior. So, what are the structures associated with the parotid gland? We have the retromandibular vein, parotid duct, facial nerve, parotid lymph nodes, auriculotemporal nerve, and the external carotid artery. These are the contents of the parotid gland. And what is the physiology? So the saliva synthesizing acinar cells. When we talk about acinar cells, these are the basic structural units of the salivary gland, the acinar cells and the ductal cells. The acinar cells are capable of producing a volume of up to 1.5 liters daily, secreted in response to parasympathetic and sympathetic stimulation. So the parasympathetic stimulation will increase the serous production, the sympathetic stimulation will increase the mucus production. Mucus in the sense the protein, which is the mucin. So it is sympathetic activity triggers the protein secretion. And the parotid glands are responsible for up to 50% of the saliva upon production on stimulation. So normally it is only 20%, but on stimulation 50% of the saliva is produced by the parotid glands. Stimulation in the sense the parasympathetic system is stimulated by means of some food or some flavor, some smell. All those things are the triggers of salivation. So all those things will stimulate the parotid gland for 50% contribution. And the saliva primarily contains the enzymes amylase and lysozyme, IgA and IgG antibodies, inorganic ions and variable quantity of mucus. The serous and mucus cells of the salivary glands contribute to the composition of saliva, and the striated ducts perform critical modification processes specifically electrolyte reabsorption and secretion, which we will see in the next seminars. So, what are all the cells which play an important role in the salivary glands? That is the acinar cells and the ductal cells. And initially isotonic saliva, isotonic is, there is the positive and negative ions are balanced. The saliva becomes a hypotonic, bicarbonates its solution as it passes through the mitochondria packed striated ducts that facilitate the ion transport. So the tonicity of the saliva changes as it is secreted, as it passes through the duct, the tonicity changes because of these ductal cells. So we will see about all these acinar cells, ductal cells in the histology part. So the function unit of salivary glands consists of acini and ducts. So acini can be composed predominantly of serous or mucuses or can be mixed seromucus type. So what are serous acini? They are the dominant component in the parotid gland and guanetmal glands of the tongue. They are polygonal in shape with dense intracytoplasmic basophilic zymogen enzyme containing granules. So that differentiates the serous from the mucus, which are periodic acid shifts reagent positive. And the mucus cells are the dominant component in the minor salivary glands of the palate and base of the tongue. They are round cells filled with mucin and contains a basally located condensed nucleus. So here in the mucus acini, you can see, I will show the pictures, the nucleus will be basally located. And it is positive for mucicarbide, Alcian blue and PAS diastase resistance, which is DPAS. And the mixed seromucus acini, they are the dominant components of the submandibular salivary gland and the sublingual glands, minor salivary glands of the lip, cheek and anterior tongue. The mucus acini with the crescent shaped demilunes of serous cells. So always this mixed seromucus acini will have a mucus acini on which the demilune of the serous cell will be present and shown in the picture. So when we see the development of the embryo, we know this is the stomatium, the ectoderm, the endoderm and the mesoderm, the buccopharyngeal membrane. And the parotid duct starts as a epithelial thickening in the cheek region. As I said, this is the cheek region where there is a epithelial budding, thickening there. So this is how, like tooth formation, we can see the salivary gland formation. There is the oral epithelium and there is a bud stage, pre-bud stage and the initial bud stage. All the things are filled with the epithelial cells. And the pseudoglandular stage. Why is it pseudoglandular? It looks like a gland, but still it is filled with the epithelial cells. There is no canalicular formation. So this is a pseudoglandular stage. And next is the canalicular stage, where you can see the proper canals which are formed. So we see the epithelial cells, the fibroblasts, and these are the parasympathetic ganglions, the blood supply, the parasympathetic nerve supply, and the basement membrane. Everything is clearly visible. This is a schematic representation. And as we see the bud stage, the pseudo-glandular stage and the glandular stage, there is a change in the epithelial cells which change into the acinar cells. And these changes happen because of these signaling pathways. We can see the TGF-beta receptors 1, 2, where the TGF-beta 1 and TGF-beta 2 go and they do the signaling transduction. So when we see here, these are all the orange cuboidal cells are the epithelial cells. So in this stage, the whole glandular structure is filled with the epithelium. And the pseudoglandular stage also, it is filled with only the epithelial cells and the mesenchymal fibroblasts. So when there is a transition, what happens? The cells which have both the TGF-beta receptors as well as both the beta 1 and beta 2 signaling, then those cells turn into the acinar cells. So that is how the transition takes place in the molecular level. So the epithelial cells are transformed to acinar cells. The acinar cells are either it can be a serous acini or a mucus acini. So when we see the, I think I showed this picture of the anatomy of parotid gland in relation to the mastoid process and the posterior part of the ramus, and this is the parotid gland as a whole. When they dissect this, we can see the lobular structures present inside, how these salivary glands develop into lobules and then they have the intercanalicular ducts. So this is a schematic representation of the mucus acini. As I said, the nuclei are placed in the basal side and they are not very clearly seen. I will show you. You can see here. So this is the serous acini, which is of polygonal in shape with a round nuclei, whereas the mucus acini are having the nucleus in the basal side. This is the intercalated duct, which is the low cuboidal cells, whereas the striated ductal cells will be tall columnar with striations. These are because of the mitochondrial activity which is happening in this area, because these play important role in the ion exchange, as I said. So these are the main parts of a salivary gland, and when we see this, this is according to the description, the nuclei is placed in the basal side. So this is a mucus acini, where we can see a serous gemellum, where there is a serous cell, acina cell, which is on the covering the mucus acini. So this is the serous gemellum, and this is the thing. And the one more important cell is the myoepithelial cells, which are responsible for the secretory function of the acina cells to push the produced saliva into the ducts. So these are the parts of the salivary gland as a histology. So this is a more detailed schematic representation. The myoepithelial cell, the serous acina cells, the intercalated ducts are cuboidal, whereas the striated ducts are tall columnar, and the excreted duct will be in squamous epithelial arrangement. The excreted duct will be like this. So as I said, the parotid gland has serous cells. So serous acina cells with the zymogen granules will appear like this, and these are the cuboidal ducts. And here we can see the serous acina cells of the parotid gland, which is polygonal in shape with the dense intracytoplasmic with basophilic zymogen granules, and the intercalated ducts is lined by single layer of cuboidal epithelium. So this is the intercalated duct with a single layer of cuboidal epithelium. And when we see the striated ducts, so these are the striated ducts. We can see the small striations in the ducts, which are the feature of the striated duct, which differentiate it from the intercalated ducts. And this one is the main duct, the interlobular duct. This is pseudo-stratified. It is looking like as if it is placed in a pan, but it is a single layer, but it is looking like a stratified epithelium. This is the interlobular duct. And there are, I will talk about the other structures which are present in the glands. One of them is the parotid gland, which contains the lymph nodes. This is a lymph node. These are the acina cells, and the lymph node is present inside the parotid gland. And the others, we can see in the lymph node there are some acina cells here. So both in the gland we get the lymph nodes, and the lymph node we get some glandular tissues. And some sebaceous cells also we can see here. And special stains are also there. I will talk about the normal inclusions within the salivary glands. The presence of sebaceous glands within the parotid gland is common and are usually found associated with the interlobular ducts, or may exist as isolated cells within the wall of the intercalated or striated ducts. And unique to the parotid gland is the presence of intraparotid lymph nodes, most of which are located superficial to the facial nerve. Often the lymph nodes may lack a well-defined capsule and merge with the adjacent salivary gland acini. So fatty infiltration of the major salivary glands is an age-induced variation. We see the parotid glands look a little huge in old elderly people because of this fatty infiltration. And what are myoepithelial cells? They are critical for the expulsion of saliva. The contractile myoepithelial cells surround acini, intercalated ducts, and the striated ducts, located between the epithelial cells and the basement membrane, embracing the secretory unit with the long cytoplasmic processes, generally spindle-shaped with the blunt-ended nuclei and elongated cytoplasmic processes. I think I showed that. So that is the thing. And regarding the special stains, we have special stains for acina cells, mucinous cells, ductal cells, as well as myoepithelial cells. So the zymogen granules of the serous acina cells are periodic acid PAS positive, and they are also low molecular weight positive for CK7, CK8 and CK19. Mucinous cells are positive for muscardine, alcium blue and PAS, that is DPAS. And ductal cells are positive for EMA, which is the epithelial membrane antigen. And myoepithelial cells for CK14, CK19 and Y19. And alpha smooth muscle actin. So here we can see the smooth muscle actin, which shows the immunostate marker for myoepithelial cells surrounding the serous acini in the parotid gland. So we can spot the myoepithelial cells around the serous acini. And this one shows what these structures with the epithelial membrane antigen, which is a ductal cell marker. So we can see the ductal cells alone marked in this staining procedure. And this is a complete cytoplasmic